All right, we're on to section 4 of chapter 4 for the McKinley text for anatomy and physiology, and we're talking about resting membrane potential. The last chapter or last section on membrane transport is a little long. This one's a little shorter for you there, but there was a lot to go in that membrane transport section, so it's just one of those things. That said, things you should be able to do with your learning in this section is to be able to define a resting membrane potential, RMP, describe the cellular conditions that are significant for establishing and maintaining a resting membrane potential, explain the role of both potassium and sodium in establishing a resting membrane potential, discuss how sodium and potassium pumps are necessary in maintaining a resting membrane potential. It really should say A. That bugs me. It somehow got in there with an RMP and an RMP. It's a resting membrane potential. That said, it's one of those things that bugs me. Plasma membrane establishes and maintains electrochemical gradient, right? It's the resting membrane potential. It's essential for muscle and nerve cell function. And the electrical charge difference that exists in the plasma membrane, there's a membrane potential, the standpoint, the potential energy of the charge difference, okay? And that resting membrane potential is the potential when a cell is at rest. The two conditions for RMP are an unequal distribution of ions and molecules across the plasma membrane, like the more potassium in the cytoplasm than in the interstitial fluid, or the more sodium in the interstitial fluid than in the cytoplasm, and that's due to that sodium-potassium pump. Or an unequal relative amount of positive and negative charges, with more positive, typically speaking, on the outside of the cell and more of a negative charge on the inside. And with that resting membrane potential, taking a look at here for the cytosol versus the interstitial fluid. So we have interstitial fluid and the cytosol here and the plasma membrane, of course. Typically speaking, you see a range for resting membrane potential between that negative 50 and negative 100 megavolts. Typical number you'll see is negative 70. We have a sodium leak channel into the cell and a potassium leak channel outside of the cell. And here's the sodium potassium pump with typically seeing what we often see is the sodium going out and the potassium coming, or the sodium going outward, inward, and then the potassium, or sodium coming inward and potassium going out. Okay, another way around. Wow. Let's try it again. So we're talking about the basically the sodium essentially going out and the potassium coming in. The most important ions, of course, are the sodium and potassium in terms of movement, depending on that electrochemical gradient. The role of potassium, it's the most important determinant and specific value of RMP, of that resting membrane potential. That potassium moves down that steep concentration gradient through the leak channels from the cytosol in an interstitial fluid, and negative charge proteins remain inside the cell. The electrochemical gradient, the positive charge outside repels the movement of potassium out, and the negative charge on the inside attracts potassium in. And of course, that potassium moves until equilibrium is reached. With sodium, it diffuses into the cells from the interstitial fluid to the cytosol simultaneous to the loss of the potassium, enters through that sodium leak channel, and then down that concentration gradient. It's pulled by the electrical gradient, and leak channels prevent a lot, as much, I should say, of that sodium into the neuron as the potassium out, and the inside becomes more positive. And maintaining that resting membrane potential, that sodium potassium pump is significant for doing that, maintaining that membrane potential. It maintains that potassium, and the sodium gradients following the diffusion, with all that sodium pumped out and potassium pumped in opposite directions against that concentration gradient. So you should simply be able to, with this fairly short section, define a resting membrane potential and explain how the resting membrane potential is established and maintained. And we'll move right into section 4.5. With 4.5 in cell communication, you should be able to explain how cells communicate through direct contact, describe the three general mechanisms of response to the binding of a ligand with a receptor. And with that direct contact between the cells, it's important for some cells to function, that direct contact between those communication between the cells. Some examples are cells of the immune system, which distinguishes the normal cell from the unwanted cells. We don't have them going after normal cells, which it can do and does happen. Sperm and oocyte, the egg with unique glycocalyx. It allows for the recognition by the sperm during fertilization. Cellular regrowth following injury, damaged tissue is replaced by that cell division in epidermis, and cellular contact prevents the overgrowth. Okay. Now, most cell communication occurs through the ligands. That's important. There are molecules that bind with the macromolecules. There are neurotransmitters from nerve cells and hormones from the endocrine cells. They're important for controlling growth, reproduction, and cellular processes. And 3 types of receptors that bind those ligands are the channel-linked receptors, and the enzymatic receptors, and the G protein-coupled receptors. So with the channel-linked receptors, they permit that ion passage into or out of the cells. They occur in response to neurotransmitter binding, and they help initiate the electrical charges to the resting membrane potential in the muscle and the nerve cells. So here we're showing the channel open, there's your ligands, that channel's closed, and of course they're open when once the ligand attaches, that opens up that channel. Okay. Enzymatic receptors, protein kinase enzymes with energy production, very vital there. They're activated to phosphorylate other enzymes within that cell. They provide the mechanism for altering enzymatic activity. So they're going to make that bind, and with that ligand here binding there, an inactive protein kinase enzyme down below. When that, of course, enzyme is going to be turned on or turned off, all depending with the binding. And the G protein-coupled receptors, they're indirectly active. They indirectly activate protein kinase enzymes. And here we get a closer look. So we have the ligand binding to receptor, which causes that conformational change to the shape and to activate the receptor. The G protein then binds the active receptor. And then here we have that GTP binding to the G protein, and that causes that G protein activation, which activates the protein, leaves the receptor. Okay. And then we have over here the effector protein and the effector protein channel. With that binding, we have the secondary messenger, and the activated effector protein then makes a secondary messenger available within the cell, which leads to protein kinase enzyme activation. Then we have the active protein and kinase enzyme, which phosphorylates other enzymes. And of course, that binding there, where the effector protein is available, is going to affect whether the enzyme is turned on or turned off. So this section, it should be 4.5, not 5.5. What are some examples of how cells communicate through direct contact? You should be able to answer that. And how do the actions of enzymatic receptors and G protein-coupled receptors differ? And we'll come back to section 4.6.